I am Jeannie Garbarino, and I have the great pleasure of serving as the director of Rock EDU Science Outreach. I see many familiar faces in the crowd and some of the new faces. How many people are here for their first time? Let me see a raise of hands. Awesome. Where's our repeat customers? Cool, keep on coming back. So it's my privilege to be here this morning and introduce today's speaker, Dr. Daniel Kronauer. But first I want to tell you a little bit more about the history of the Rockefeller University. So it all began in 1901 when the beloved grandson of John D. Rockefeller Sr., Jack Rockefeller McCormick, contracted scarlet fever and died a few months before his 4th birthday. You can see a picture of young Jack right here with his family. This was a major blow to Mr. Rockefeller, and it inspired him, along with his financial advisor, Mr. Gates, to do something that had great significance for humanity. Mr. Rockefeller realized that more really should be done to advance medicine and to prevent the spread of disease. And to make real progress, he knew that scientists needed to understand the disease process at a very basic level. So he founded the Rockefeller Institute of Medical Research, a dedicated biomedical research institute that quickly became a leader in the fight against disease and in the effort to advance scientific knowledge. In 1955, the Rockefeller Institute expanded its mission to include doctoral education, and 10 years later became known as the Rockefeller University. And here is a picture of the first graduating class from Rockefeller's Ph.D. program, along with the then President Detlev Bronk, who is also seen here with David Rockefeller looking at the model of the building that we are sitting in today. So this is before it was built. Now, despite our label as a university, the students on this campus have already received their undergraduate degrees. So we don't actually have undergraduates here, and had come to Rockefeller to earn their PhDs or MD PhDs. Like most scientific trainees across the country, students here pay $0 in tuition, and they also receive a lot of support in terms of subsidized childcare, subsidized housing, and other types of support to make sure that the work that they are doing is of the highest quality and their personal lives are supported as best as possible. There are approximately 200 graduate students here on campus each year, working with more than 600 scientists who range from postdoctoral fellows to the professors who head the university's 81 laboratories. Over the years, the work done at Rockefeller has been recognized many, many times throughout the world. 25 Rockefeller scientists have been awarded the Nobel Prize, more than most countries, and 23 have received the Lasker Award, also known as the American Nobel. I actually need to update this slide. It's happening so fast that my slides don't even get updated because the scientists here are so fantastic. It's pretty amazing to note that 3 Rockefeller scientists have been honored with these awards in just the past 2 years. Firstly, we have Charlie Rice, who's an amazing scientist, and he heads the university's Laboratory of Virology and Infectious Disease. He received the Lasker Award 2 years ago for his pioneering contributions to the development of the cure of hepatitis C, a virus that infects 170 million people worldwide. And I am very pleased to also say that Charlie Rice is a big supporter of science outreach and has trained many high school students as they have come through Rockefeller Gates to learn about the process of science. We also have Michael Young, who's the head of the Laboratory of Genetics. He received the Nobel Prize in 2017, the Nobel Prize in Physiology or Medicine, for his pioneering discoveries about genes and molecular mechanisms that underlie circadian rhythm, your body's daily biological clock. Interestingly, I talked to Mike yesterday, and he told me that he was jet-lagged, and this is a phenomenon that he himself is still looking to figure out. So even the best scientists need to ask more questions. And just a few months ago, David Alice, the head of the Laboratory of Chromatin Biology and Epigenetics, received the 2018 Lasker Award for his discovery that chemical modifications of histone proteins provide key signals to turn genes on and off a discovery that has proved critical to understanding a wide range of diseases, including autism, congenital heart disease, and cancer. So he works on epigenetics. You must hear about that in your classes a lot. Yeah. Some of the teachers in the audiences might have attended Talking Science in 2013 when Dr. Ellis was the featured speaker. Do we have anybody who was in that audience? Hey, good, that's amazing. So working at Rockefeller for me, I've been here since 2008. I came here first as a postdoc, studying under the mentorship of Jan Breslow. I worked in metabolic biology and biochemical genetics. And I just constantly came to work like amazed that how did I get here? It's so wonderful to walk through these gates every single day. And it's like being in scientific Disney World. And I was talking to another colleague who said that here at Rockefeller, if you dream it, you can do it. It is an incredible, special place. And scientists have indeed done it, as I've just described with some of the awards, but there's so much more going on. But when you see the finished product, such as the chemical structure of an antibody, or the important immune functions of dendritic cells, and if anybody's interested what that looks like, I'm wearing one of the first images on my lapel here that Ralph Steinman took of a dendritic cell. Come say hello, and I'll show you my pin. And we've also had scientists here who took the first pictures of the birth of HIV particles in living cells. Like, that was just astounding. But again, the finished product does not necessarily tell you about the process involved in making these discoveries. I think it's really important for us as a society to appreciate that scientific discoveries take time, sometimes on the orders of years or even decades, and that it typically involves constant learning from failure and iterating on your strategy. Sometimes that iteration involves using super fancy equipment, like our amazing cryo-electron microscope, which allows you to view samples at the atomic level. And seriously, I go in there all the time because it's so cool and it's next to my lab. And I go in there and there I see an atom of gold with my own eyes. I mean, can you imagine seeing an actual atom? It's the most mind-blowing thing I've ever experienced. We also have these crazy cool state-of-the-art microscopes that in our microscopy resource center. And here, it allows you to take these really cool 3D pictures. So this is a 12-day-old mouse embryo that was captured by my colleague, Gist Croft, in the Brivenloo Laboratory for Stem Cell Biology and Molecular Embryology. Now, there's no sound on this, but Gist often sends me these videos set to like David Bowie music. It's really cool. I thought I'd spare everybody my love of 80s music. Or not, I don't know. I hope you guys love 80s music because it's the best. But other times, I'll let you just see this for a second because I just, I'm pretty sure those are the kidneys, and there's some information on the circulatory system. It's just really incredible. But other times, other scientists rely on everyday items or hacks in their research. So we have scientists who kind of fashion little iPhone holders out of styrofoam so they can take pictures on their microscope. I love this. So this is from a postdoc in the Vassal Laboratory of Neurogenetics and Behavior. Leslie talked last year at Talking Science, so many of you might be familiar with some of the work that she's doing in her laboratory. And basically, Laura uses different color glitter to identify her various experimental mosquito groups. She shared with me, I'm just gonna play this because this is so impressive. She shared with me that this technique has a very technical name. It's called shake in a bag. So basically, when she has different types of genetic experimental groups, she'll put some in it, or maybe she's adding, some sort of pressure to one sample. She'll put them into different bags with different color glitter and just shakes them so that she can tell them apart in their experiments. So that's really fun. And then apparently, let's get here. Who recognizes what this thing is from? It's a Baskin-Robbins spoon. And this is the choice tool for Nicholas Svedic from the Zhao Laboratory of Evolutionary Genetics and Genomics for his fly egg-laying experiments. He explained to me that the spoon substrate is ideal to take groups of eggs in or out of the vial to count and look at newborn larvae. And this is actually, the green is food colored, so he uses food dye to color fly food so that he can have the contrast for when they have the larvae. So it's really cool. Another cool hack that people use is your Cuisinart food steamer that you get out of Target. This is the best piece of equipment for doing heated slide incubations. And Duplo blocks are apparently the best for novel recognition test in mice. And this is from the Strickland laboratory. And this is maybe my favorite, Manic Panic. How many of you have ever used Manic Panic? Yeah, a couple, right? So Manic Panic UV reactive hair dye helps our researchers in the Fuchs Laboratory of Mammalian Cell Biology and Development study the adult stem cell niche in mouse hair. And so I have one more set of examples, and these are in honor of our featured speaker today. Ant researchers, like our colleagues in the Kronauer lab, whose work I'll introduce in more detail in just a minute, maintain ants' nests using plain old Tupperware and plaster of Paris. So I was really shocked when I saw that. And ant food is store-bought honey, although I did learn that they really like tuna fish. So that was kind of a cool fact. I think I'm gonna be really good at Jeopardy one day because I have all this stuff stored in my head. Ants within a colony, so this is a really cool video. I'm just gonna fast forward it a little bit for you. Let's start from here. Okay, so what you're seeing is how many people have used the Snapchat geofilter today where you saw the ants with like the little colors on their butts, right? And so this is how we put that, or ant researchers put the color on the ants. And so basically they take a pencil eraser and gently place the ant under a little tiny elastic and use a needle to deposit paint. And this is paint that is from plain old paint markers that you just buy, from Amazon or Target or something like that. And so I think these are really cool examples of how, you don't necessarily need cryo-EMs and crazy fluorescent microscopes to do science. You can do science with pencil erasers and Tupperware boxes and Duplos, so science is really accessible, and I think it's a wonderful opportunity for people like you to think outside the box or inside the box, I don't know if you're using a box, and do the science that makes sense to you. And so I wasn't kidding when I called this place Scientific Disney World. But science does not happen in a vacuum, and we believe that science for the benefit of humanity, which is Rockefeller's ethos, also includes sharing our science with students and teachers. In fact, Rocky2U Science Outreach works with year-round students and teachers to present fun, authentic science research experiences to thousands across the city. Whether through our Science Cafe, Rocky2U Presents, which is happening, if anybody's interested in joining us again next Friday, it's taking place in Carson Family Auditorium at 4:30 p.m. on January 18th, or participation in our mentored research programs such as Lab Jumpstart or the Summer Science Research Program, we hope to have you back with us. But one event I look forward to each year is Talking Science. Talking Science is a very special part of the tradition of Rockefeller scientists giving back. This lecture series began in 1959 when it was presented as an annual Christmas, then holiday lecture on science for high school students. It was established by Alfred E. Mirsky, a biochemist at Rockefeller, who was one of the pioneers in unlocking the secrets of the cell nucleus. Dr. Mirsky modeled the lecture on a popular series of science talks for young people established in London in 1825 by Michael Faraday, who is considered one of the greatest experimenters in the history of science and one of the greatest communicators. Today, we are pleased to welcome students from more than 50 schools from across the 5 boroughs of New York and beyond. Thank you for spending a precious Saturday in pursuit of scientific knowledge. So how about you give yourselves a round of applause for getting up early? Yeah? I know students aren't getting enough sleep these days, so thank you. On behalf of the university, I would like to acknowledge Merck for their generous support in helping us to present today's exciting program. Now, it's my pleasure to introduce Dr. Daniel Kronauer. Daniel was born and grew up in Germany. He has been fascinated by insects since he was in kindergarten, when he would turn over rocks just to see what kind of insects were living underneath. As a teenager, he became fascinated with tropical biodiversity and initially wanted to become a tropical ecologist and conservation biologist. He began traveling in the tropics in his last year of high school, and by the time he went to college, 2 years later, he had been to the American, African, and Asian tropics. And this is a fun fact that I learned. Daniel won one of his early trips to Cuba in an Elvis Presley karaoke contest held by his local radio station. One day, maybe we can get you. Not today, though. Daniel received a degree in biology from the University of Würzburg, followed by a Ph.D. from the University of Copenhagen. He was elected to the Harvard Society of Fellows as a junior fellow in 2008. He joined Rockefeller faculty in 2011, where he is now the Stanley S. and Sidney R. Shuman Associate Professor and head of the Laboratory of Social Evolution and Behavior. We have the privilege of having Mr. and Mrs. Shuman with us in the audience today. And on behalf of the university, I would like to thank you both for your tireless support of Daniel, the university, and the mission of science for the benefit of humanity. So thank you so much. Here at Rockefeller, Daniel studies ants and the sophisticated colonies in which they live. By nature, ants are social and prefer group living, and their colonies are highly organized around different tasks. I forgot to advance so you can see this amazing microscopy image of an ant. You may have recently seen articles about Daniel's research in the New York Times. If not, they are available and can be easily found on the Talking Science website. Today, Daniel will discuss the research he conducts in both the laboratory and in nature. This morning's topic, studying complex ant societies in the field, will focus on army ants to illustrate the organization of the most complex insect societies. Massive colonies that can contain several million individuals. Many of the images that you will see during this lecture are photographs taken by Daniel himself. After lunch, Daniel will cover a talk about studying simple ant societies in the laboratory. In particular, he will focus on the clonal raider ant, which is a close evolutionary relative of army ants, but lives in a much smaller and simpler colony. He will discuss his lab's efforts to develop and use the clonal raider ant as a laboratory model system to understand the fundamental organizational principles of insect societies and to study the molecular and neurological underpinnings of social behavior, or neurobiological underpinnings, excuse me. He will also illustrate how laboratory experiments under precisely controlled conditions can complement field studies and provide insights into basic questions in evolution, genetics, and neuroscience. Please welcome Dr. Daniel Kronauer to the podium.
All right. Thank you, Jeannie, for the kind introduction, and welcome everybody to Talking Science at Rockefeller University. I hope you have a fun day hearing and learning about ants and seeing the campus and getting some feeling for what we're up to here. So I've split up my talks into 2 lectures, and this morning I'm going to talk about work that we do in the field, and mostly I'll focus on work that we do in the neotropics in Costa Rica. And then in the afternoon, I'll talk about some of the work we do on ants in the lab under much more controlled conditions, as Jeannie already said. And of course, what we're all after by doing this kind of research or by studying biology is that we want to understand some of the principles that govern life. And now in biology, it's a little bit different than, for example, physics, where people talk about more universal laws. In biology, there's very little in terms of universal laws that don't really have exceptions, other than, of course, that biology is ultimately bound to the laws of physics. But what I'll try to convey a little bit in my talks today is that there's still some pretty broad principles that you can understand. And of course, by studying biological phenomena, you can then try to understand when these principles apply and what causes exceptions from these principles. But I wouldn't necessarily call them laws, at least not if you're thinking about physical laws. And what I'll also try to show you is that these kind of questions, they can be asked at all levels of biological organization. So you can ask them at the level of genes, you can ask them at the level of cells, you can ask them at the level of whole multicellular organisms, you can ask them at the level of societies, of insect societies in my case, and you can even ask them at the level of whole ecosystems. And this first talk, I'll focus mostly on the structure of ecosystems, and I'll just give you some examples that involve ants. And then in my afternoon talk, I'll focus on the structure of societies and how those arise from interactions between multicellular organisms. And I'll continuously try to draw parallels to organizing principles that underlie how cells organize in multicellular organisms. And I hope to convince you that there are some very striking and interesting parallels that again point to these general principles that underlie the organization of life. So that's one of the things I'll try to convince you of today, and I hope will be one of the take-home messages for my talks. Okay, did I forget anything? Yes, ants are amazing. That's the other take-home message that I hope you'll get from today, but we'll see. That's hopefully an easy take-home message. And I'll start with this quote when we think about ecosystems from Charles Darwin, very famous book on the origin of species. That's kind of the foundation of evolutionary biology. From 1859, where he writes in his closing paragraphs, "It is interesting to contemplate a tangled bank clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through damp earth, and to reflect that these elaborately constructed forms, so different from each other and dependent upon each other in so complex a manner, have all been produced by laws acting around us." So this is this famous idea of nature as an entangled bank, where everything is kind of interacts, and is in some way or another directly or indirectly dependent on each other. In more modern terms, we can think about an ecological ecosystem as what we call an ecological network, where, again, everything is kind of connected. And this network idea is very prominent in biology. So you can think of ecosystems as networks, but of course you can also think about, say, gene regulatory networks. You can think about metabolic networks in a cell. You can think about neural networks in a brain. And there's some, at least at a very basic level, there are parallels about how these networks are regulated. So it's very helpful to think about different organizations or different levels of biological organizations in the framework of networks. And of course, these networks can be very complex, but it's very helpful to understand some of the basic organizational principle of these networks, if you want to understand how these networks work and how they respond to perturbation, for example. And if you go out into the rainforest, for example, in Costa Rica, as we will in this morning lecture, of course, it's very difficult to study this whole ecological network at once. It's essentially impossible, but you can study smaller sub-networks, maybe that are embedded in this broader networks. And for example, these can be networks of mutualistic organisms, so organisms that interact and that benefit each other. And I'll show you some example using ants later. You can study predator-prey interaction networks, and that's one of the studies I'll talk to you about in this morning lecture. Or you can study, for example, host-parasite interaction networks, and that's going to be the second example I'll talk about. And so first, of course, you start by simply trying to figure out who interacts with whom and in what capacity, of what nature are these interactions. And then once you start to understand these kind of dynamics, that can help you understand how species can coexist. Why do you have, say, in a rainforest like Costa Rica, why do you have 400 different ant species in that rainforest, and how is that even possible? You can start to ask, how do these species co-evolve? How do they evolve adaptations to coexist with each other? You can also ask very basic questions about what regulates the population size of a given species. So who's preying on that species, what does that species feed on, and so on. And once you understand these interactions, it gives you some predictive power as to how an ecosystem will respond to perturbations. Like, what do you have to do to an ecosystem to, say, push it in one direction or another, and under what circumstances might this ecosystem go extinct and the whole network breaks down. So just to give you an example, now I spend some of my weekends in upstate New York, and I try to flip rocks and look for ants with my son now, so I'll try to pass that on. We'll see how that works. But so when you're in upstate New York, over the last years, it has been more and more prevalent to contract Lyme disease, which is a disease that's transmitted by ticks. It's a bacterial disease. And it turns out that the tick population is very dependent on the population density of things like deer that carry the tick. These are deer tick, but the ticks are also carried by small rodents like chipmunks, squirrels, white-footed mice, and so on. And of course, the deer population is so high because there's very few predators, for example, if you're in the Catskills. And these are all kind of non-trivial interactions, unless you understand them, you don't have a good feeling for why suddenly there's such a prevalence of Lyme disease in upstate New York. But once you understand these interactions, you could think about if you introduced wolves, say, in this ecosystem now, maybe that would control your probability of getting Lyme disease, right? Which is not a trivial connection to make necessarily. So what does that have to do with ants? Why do I focus on ants in this particular context? Well, one reason is that ants are extremely diverse. So there are over 14,000 described different ant species, and there are probably many more that we haven't even discovered yet. So they're very diverse, and they're pretty much everywhere. You can find ants in almost all terrestrial ecosystem, maybe other than Antarctica. And the ants are extremely ecologically dominant, so they play very, very central roles in structure and ecosystem and interacting with other organisms. And again, I'll show you a few examples in a minute. So if you took all of the ants in a tropical rainforest, like for example Costa Rica or the Amazon, and you took all the large animals, all the vertebrates, and you put those on a scale, you would find that the ants actually outweigh all the big animals in these ecosystems. So they're extremely, not only extremely diverse, but they're also extremely dominant. There's not only many different types of ants, but there's just a lot of ants in these ecosystems. And again, this is a picture from our field site at La Selva Biological Station in Costa Rica. We walk along these muddy paths throughout the rainforest, and we look for ants. And the tropics, they've always been, I think, a great inspiration already for these early naturalists like Darwin, because this is really where terrestrial biodiversity is the highest. And this network of the tangled bank is particularly tightly woven. So some of these ecological and evolutionary principles that we are after, they are particularly apparent in the tropical rainforest. And I'll start by just showing you some of my favorite ants at this field site in Costa Rica. And I'll try to give you examples for these different types of interactions that different organisms have in that rainforest. This is an ant species of the genus Azteca. And these guys, they're mutualists with plants, Cecropia trees. And what happens is that the ants, this is the nest entrance hole. And so the ants live inside the hollow stem of the Cecropia tree. And the Cecropia tree, on top of that, it not only provides housing for the ants, but it also provides food bodies at the base of these big leaves. And then the ants, in turn, they defend the Cecropia tree. They're extremely aggressive. So if you happen to walk through the forest and you by accident, you lay or you put your hand on a Cecropia tree like this, you immediately get swarmed by the ants and they bite you. And it's pretty painful, actually. So they defend the tree against herbivores and so on. So these trees, they really, as long as they are occupied by ants, they really look pristine. And as soon as the ant colony dies or it gets removed experimentally, these trees get overgrown by other plants and they get eaten by insects. So the ants are very good at defending them. And if you slice a tree like this open, you see this is inside the nest chamber of the ant. These are the larvae of the ant, the young. And all these white little globules, these are the food bodies that the Cecropia tree produces at the base of the leaf. So the ants harvest them and then they bring them inside the nest chambers. So it's this very tight-knit mutualistic interaction. And there are many Azteca ants, there are many Cecropia trees, and there are also many other plants that have similar associations with other types of ants. This is another really important ecological interaction between an ant and a plant. These are leaf-cutting ants in Costa Rica. They're actually major agricultural pests, and they can defoliate entire trees overnight, a big colony of these leaf-cutter ants. And here, this is where the name comes from. They cut the leaves, and they bring them back into their nest chamber. And in the nest, these ants are also known as fungus-growing ants, because what they do with the leaves is they cultivate a fungus that grows on the leaves. And then the ants actually don't eat the leaves, but they eat fruiting bodies from this fungus. And this is a picture of an incipient leafcutter ant colony. What you see up here is the queen, so she's massive. These are her first young workers. They're tiny in comparison. But this leafcutter ant queen, she can live for several decades, and she will produce like millions and millions of offspring during her lifetime. So this is a small starting colony, but this is a picture where people excavated a mature leafcutter ant colony, and they had to pour 8,000 tons of concrete into this nest to kind of solidify it, and then they excavated it. So you can see that these kind of nests of leafcutter ants, they can really reach enormous dimensions. And when you zoom out, this is now satellite images from this area in the Paraguayan Chaco, and you see that these white dots, these are actually leafcutter ant mounds. And so here you can really, I think, and these little, these dark green dots, they're palm trees. So I think from something like this, you can really appreciate what kind of ecological impact an ant species can have, right? What kind of impact the species has in structuring the ecosystem. So this is actually now the first depiction of, or one of the very first depictions of leaf-cutting ants. This is by a German naturalist, Maria Sibylla Merian, who traveled to Suriname in South America around 700, and then she published this book in 1705 back in the Netherlands. And you can see here, she depicts leaf-cutting ants in this typical fashion. They're like up on the tree and they're harvesting leaves. And what I really like about these early depictions by Merian is that unlike most of the other naturalists around this time, Merian already places all the animals and the trees and the plants that she encounters in this kind of network framework, right? So she shows how these different species interact. Like usually you look at books by early explorers and they have a white page and there's like a jaguar in the middle and that's it, right? Versus in Merian, she has, by almost like 150 years, or more than 150 years, she seems to like anticipate this notion that Darwin later had of the ecosystem as an entangled bank. And what you'll see here is that, unfortunately back then, Merian, she kind of confounded the two superstars of neotropical myrmecology, of the neotropical ant world. So the leaf-cutting ants up here, but down here you see ants that are actually attacking some kind of insect on this guava tree. And those are actually two different species. And in the description that goes with this plate in Merian's book, it also becomes clear that these two species have very different biology. She just kind of paints them as the same thing. So these guys here are the leaf-cutting ants that I've already showed you, but these guys down here, these are actually army ants. So those are the second superstars of ant biology in the neotropics. And those are the guys that I'll focus on during my morning lecture. So army ants are really top arthropod predators in tropical ecosystems around the world. This is a species that's particularly stands out in the neotropics. The species is called Aceton bocellii. And here you can see a swarm rate that basically goes through the forest leaf litter, and they even climb up the tree, these ants. And here they overrun a katydid. So in numbers, they can overwhelm very large prey insects. But army ants, again, they inhabit the tropics around the world. You can see here some species even go up into the southern US, the kind of more subtropical regions. They go down into Argentina even. But the center of the distribution and the most intense ecological impact they have in the tropics. This is just now a more schematic depiction of what a swarm rate of this one species, Aceton bocellii, looks like. So they form these carpet swarms on the forest floor, and the swarm front can span on the order of like 20 meters across, and it's about like 2 to 3 meters deep. And so you have this carpet of hundreds of thousands of ants that moves through the leaf litter on the forest floor, and then behind the swarm front, the ants collapse into these columns until they reach a base column that then connects the swarm front to the bivouac. And the bivouac is kind of this army parlance in terms of army ants that describes the army ant nest. And I'll show you what the army ant nest looks like in a minute as well. So this is what it looks like at the swarm front. Of course, that's difficult to take a picture of in a rainforest environment, so this is a little bit zoomed in. But you can see that there's just this carpet of ants that now moves through the forest. And you can see that all the ants, or most of the ants, have their mandibles wide open, so they're very aggressive and they're really looking for things to attack. And they do that with basically everything that falls into their path. This is another example of an orthopteran that's basically just getting overrun by the ants. This is what it looks like when the attack first starts, and this is just a few seconds later. The ants form this big ball on top of this orthopteran. And just to illustrate what kind of ecological impact these ants have in the tropical rainforest, what you see here is a swarm rate of aceton ants going up a tree. You can see the ants here on this white background of this trunk root of the tree trunk, of this buttress root. And these swarm rates, they're so potent that they attract very specialized birds. What you see here is that's called an ocellated ant bird. And there's a whole guild, like several species of birds in tropical rainforest that basically obtain all or at least most of their food at aceton buccelii ant swarms. And what they feed on are these insects that get flushed out by the ants from the forest leaf litter. And this species here, this ocellated ant bird, they're so specialized that they actually can keep track of different army ant colonies as they move through the forest. So they know exactly where in the forest the different army ant colonies are and where they're hunting at any particular moment. So if this swarm here dies down, they just fly off and they go to a different swarm. And they actually go in the early morning, they show up at the ant nest and they wait until the ants start hunting. So they have memory as to where the different ant colonies are. And you can also show that if you have fragmented forests and the ants decline in abundance, so they even go extinct, these ant birds will also go extinct in those forest fragments. Now, when they attack these very big insects, of course, they are too large to transport for a single ant. So these army ants are very good at cooperative transport. What they do is they cut up the big prey insects into smaller pieces. And here you can see them carrying a big leg from a katydid back to their nest. And they all kind of work together to move this big prey item. But in fact, it's only a few species of army ants that attack large insects, and in some cases even small vertebrates. Most army ants prey on other ants. And this is a picture, again, of the same species, aceton buccelii, as they attack a nest of carpenter ants that's nesting inside a tree trunk. And you can see here, this is the nest entrance. And here as well, the army ants, they basically just overrun the colony. Initially, the carpenter ants, they can defend the colony. They recruit their soldiers to the nest entrance. And this onslaught can last for a couple of hours, but eventually, in this case at least, the army ants eventually got into the nest. And here you can see them drag out the carpenter ant workers. So you can see some here, and here, up here. And then they overrun the nest, they get all the brood out, and in many cases they kill the colony. Here's just a close-up of how they fight with other ants. And again, these other ants, they can be much larger than the army ants in individual size, but of course they also have to crack large colonies of ants that are very well defended and usually not accessible for other types of predators. Okay, so now I'll shift gears a little bit and I'll talk a little bit about this community of army ants at this one rainforest site at La Selva in Costa Rica. So already from this one genus, this is the species I showed you a few pictures of now, aceton buccelii, but even of this one genus, there are actually 6 different species. And there are quite a few more army ant species from related but distinct genera. So in total, there's about roughly 20 species of army ants at this field site in Costa Rica. And so what I'm going to talk to you about is one study where we looked at this predator-prey interaction network of army ants and their prey. And a second study where we looked at the interaction network between army ants and social parasites that infiltrate army ant colonies to take advantage of the ants. And these parasites, they usually feed on all the resources that they can find within the ant colony. So they feed on the prey, they feed on the larvae of the ants, in some cases they even prey on the adult ants. So if you just sit at one of these army ant raids and you just collect a few of the prey items that they retrieve from the swarm front, this is kind of what you get from an aceton buccelii raid. And this is, of course, now not very systematic. This is just a few prey items that I collected over maybe something like a 2-minute interval, and then I took them back to the lab and I photographed them. But you can already see that the prey that an aceton buccelii colony brings in is very diverse, right? So you see that they also, to a large extent, prey on ants. These are all ants. Here's some ant pupae, so they're just undergoing metamorphosis. But you can see that these are a few different ant species in this prey sample. But then there's also a lot of different insects, right? There's this big centipede, there's a beetle, and then there's all kinds of different parts of other insects that are, of course, very difficult to identify because you only collect them in parts. The ants have already chopped them up. This is now what a similar sample looks like from a second species, Aceton hamedum, which is quite closely related to Aceton buccelii. But you can see that here, basically all the prey items you retrieve in this 2-minute intervals are ants. There's some different ants. There's a lot of these kind of ants. These are actually a species of leafcutter ants. But then there are other ants in there too. There's these like tiny ants. So they basically prey on ants. This is now the prey sample, again, collected over about 2 minutes from a third species, Aceton vagans. And in this case, all of these ants that you see on this picture, they are from the exact same species. These are male ants, so they look a little bit different, but they are the males of these workers. And down here you can see the queen. So basically what it seems like here, this species has just plundered one ant colony of one species. So now we can do this a little bit more systematically, and we can ask what is the food spectrum of the different army ants in this rainforest? Like, what do they eat? How specialized are they? Which species have very broad diets, or are there species that have very broad diets, and other species that are very specialized? And that, of course, will allow you to figure out how these different army ant species in that rainforest habitat can divvy up the available prey, and how they manage to coexist, right? And so the way we approached this problem is we did very systematic sampling, so not just like sitting there for 2 minutes and collecting random items, but really doing it in a systematic way from a defined number of colonies and in a defined sampling protocol. And then we followed this by a fairly large-scale DNA barcoding, and I'll show you how this works. So if you want to identify all these specimens that you collect, and now we're talking about hundreds and hundreds of specimens by eye, that's very difficult. First, because it takes a lot of time, but also, as I showed you in these pictures, some of these prey items you can't really identify by eye. And the reason is that some of them come in small pieces, so you get like a little part of a leg of something and you don't know what it is, or it's some egg or a larva of an ant species, and these are also essentially impossible to identify. So in most cases, we start with an unknown organism, and what we do is we extract DNA, we bring it back to the lab, we extract DNA, then we perform PCR, so we amplify a certain region of the genome, which we call a barcode, and then we perform DNA sequencing. We perform another round of PCR where we incorporate, instead of the normal DNA bases, we incorporate DNA bases that are labeled with a fluorescent dye. And then this can be read out on a gel or on a capillary. Maybe you've heard about how this works, but essentially you can really, from this chromatogram that you get, you can really read the genetic sequence at that particular locus in the genome. And once you have that, that acts like a barcode. And you can compare it now back to a database where you have barcodes that are matched to known species. And if you're lucky and the species you're trying to identify is contained within that barcode library, you might get a very good match. And then you kind of know what species your sample comes from, what the barcode matches to. So in some ways, this is a little bit similar to forensics, right? Where you have like a DNA sample, but you don't know who it's from. So you do in forensics, of course, you do a very fine-scaled barcode, but then you can assign that sample back to who it came from, as long as that person is in your database. And again, what this allows you to do, this approach, you can identify specimens from small fragments that you can't otherwise assign. You can identify different life stages, so you can basically reconstruct the whole life cycle of an organism, or in our case, with the prey items, we can say what species a little larva comes from. But it also helps you to delineate species boundaries. So sometimes you get DNA barcodes from two specimens that you think are extremely similar, but then it turns out that in terms of their genetics, they're actually quite distinct. And in those cases, and that will become important for the second part of the talk, with some additional analysis, you can actually get some very fine delimitation of species boundaries. What is one species and what is a different species? And that's, of course, extremely important if you're interested in biodiversity and the specificity of how species interact. So this was a project that was spearheaded by Christoph, who was until very recently a postdoc in the lab, and a German master's student, Phil. And they spent a lot of time in Costa Rica sampling prey items from army ants, and they ended up analyzing over 3,000 prey items from 11 different army ant species at this one site. And so we're missing quite a few species here, and the reason for that is that a lot of army ants actually hunt entirely in the soil, so you hardly ever see them. And then, of course, it's very difficult to get any kind of information on them. And it turned out that from all these systematically sampled prey items, actually 98% were other ants. We could assign to other ant species. So it turns out that army ants are really mostly ant predators, and it's only a few species that really take other types of prey. They also mostly prey on ant brood, so 87% of these prey items were ant brood rather than adult ants. And we could see that collectively, this army ant community that we studied, at least these already these 11 out of the 20 or so species that occur there, they rated a total of 129 ant species, and that's about 30% of the local ant fauna. And again, this is kind of a minimum estimate, of course, because first our sampling is limited, and as I said, we only sampled about half of the army ant species. And now you can use all this data and you can plot it in an interaction network. And so on the left side here, you see all the different army ant species that we sampled, and the width of these bars corresponds to how many prey items we got from each species. And of course, some species are sampled much more thoroughly than others because some of these army ant species are very rare or very difficult to encounter. And then you can draw these connecting lines to the prey species, right? So every bar here on this right side represents a different ant prey species. And here's the name of these different species. It's, of course, way too small, so you can't really see what it is. And you see that these non-ant prey, so other insects, they make up a very small fraction of these preys that we sampled. But you can see that among Eceton-Bucellii, the prey spectrum of this particular species, non-ant prey actually makes up some significant proportion, and that almost all of the non-ant prey in our sample had been collected from Eceton-Bucellii swarm raids. But so what you can also see is that some ant species really seem to be preyed on only by one army ant species, and other ant species are preyed out by more than one, and sometimes many army ant species. And on the other side, you can see that there's quite some division in the prey spectrum between the different army ant species, right? So there's some overlap between some of these army ant species, but overall that overlap is quite limited. So we see very high prey specialization in this ecological interaction network, and very small prey niche overlap. And of course, we can quantify this from a network like this. But we can do additional analysis with this kind of information. So for example, we know where these different ant species are nesting. So we know whether they, for example, usually have the nests up on trees or whether they are subterranean in the soil. And so just from sampling prey that we collect from these raids, when there's a column of ants, of army ants passing across a trail, for example, we can tell where they've been. And so it turns out that some of these army ant species, they really only take prey that lives up in trees. And of course, usually you don't go up high on a rainforest tree and you conduct your ant surveys there. But now we know that these species obtain most of the prey from high up on rainforest trees, versus others basically only attack ground nesting species. So they never climb up the trees, apparently. We can also look at temporal differentiation, like how the army ants split up the prey spectrum at the site over time, and we see that some of the army ant species, they're almost exclusively active during the day when they raid, and other species are exclusively active at night. So there's both the spatial differentiation in niche, and there's this temporal differentiation in niche among the army ants at La Selva. Okay, so just to summarize this part of the talk, what I've shown you is that any given army ant species affects a limited number of prey ants directly. That some army ants are much more specialized than others. You saw that there's some that really prey only on very few ant species, whereas others, like Eceton-Bucellii, preys on many ant species and even other types of insects. But that as a community, the army ants affect the majority and maybe actually all ant species in that ecosystem. And those are only the direct effects. What I've also shown you is that army ants affect other species indirectly. At least I showed you this example of the bird that's following the army ant swarms. There are many, many more organisms that are indirectly dependent on the ants. So you can, I showed you in my intro slides the ecological impact that different ant species have on the ecosystem other than the army ants. And of course, all of these species are top-down regulated by the army ants as predators, right? So these effects that army ants have directly on the prey ant community then has downstream cascading effects on all other kinds of organisms, like, for example, the trees that were inhabited by the ants, right? So now, of course, we can think about how to follow up on these kind of first insights we got from this one side, and we could move on and we could start to look at other sites, right? And we could ask, how does the prey spectrum of a given army ant species change as you change the composition of the army ant community? For example, is there competition between different army ant species that they exclude each other from raiding particular ant species? And if you remove one of them, does the other ant species then broaden the prey spectrum and start to raid additional ant species that are in the ecosystem? Or what happens if you remove particular prey ants from the community of prey ants? How does that affect the rating pattern that you see among the different army ant species. So now you can, by comparing, it's very difficult, of course, to do direct experiments where you remove, say, an army ant species entirely from an ecosystem, but you can do something that's maybe the second best option. You can compare different ecosystems that differ in their community structure, and you ask what kind of patterns are general and which ones are idiosyncratic to any particular site. So now, kind of as a segue into the second part of the talk, I'll show you some of these other amazing collective features that you see in army ants. And one is that army ants are really masters of living architecture. So what you see here is a bridge that, again, this is S. tombocellii, this one species. This is a bridge that the ants construct out of their own bodies. This is along a raiding trail. And so you often see this where there's gaps in the leaf litter. The army ants construct these bridges. And then when other ants come with prey items, they can just walk over these bridges. So that makes just the traffic on the trail much more efficient. This is a small bridge that I've photographed here, but I'll show you a video of what this looks in the extreme case. This is now a colony of S. tombocellii that's attacking a wasp nest that you see here in the right corner. And they've basically constructed this massive suspension bridge. And you see that the ants travel over this bridge, right? The whole bridge is made up of live ants. It's not like a vine or anything that they travel on. These are all live ants. Here you see some of the desperate wasps flying in the background. And here you can see some of the ants coming back with wasp brood when they carry these large white items. Right? This is like on a research cabin in Costa Rica, actually. So now if you think that you have an ant invasion in your house, you can put it in perspective. That's what a real ant invasion looks like. So here they are at the wasp nest. And so this is a picture I took of one of these bivouac nests, which is another great example of this collective architecture and the collective behavior that they have. In my intro slide, I showed you, that was on the right side, I don't know if you remember, there was a picture of these ants forming chains. And these chains, they often use as scaffolds, and here they use it as scaffolds for one of these bivouac nests. And so here, this precise structure of this bivouac nest basically emerges from the interactions between the individual ants and their environment. So here, they built the bivouac nest against a plant that had these two little stems, and then there's a larger stem here, and that gives rise to this kind of cathedral-like looking bivouac nest. But depending on where exactly they construct it, it can look quite different. And so now the reason that the ants have a nest like this, unlike, for example, the leafcutter ants that I showed you in the beginning that have this massive permanent underground nest, is that the army ants, they are nomadic. And basically what they do is that at night they relocate their whole nest. And probably ultimately from an evolutionary point of view, the reason why they have to relocate the nest is that they're just so efficient hunters that they basically deplete the local food resources during the daily raids. And then at night, they basically relocate to new hunting grounds. So they're completely nomadic, and that's why they have these very dynamic nests that they can easily assemble and then disassemble again. And this is what an army ant immigration looks like at night. Now they're carrying not prey items, but they're carrying their own larvae. Here you can see another cool kind of collective feature is that they build these flanges when the emigration column passes like a very narrow thing, like this little branch here. So the ants that are actually carrying the brood, they don't slip off. And they do these emigrations in cycles. So the cycles, the colonies of at least this particular species and many related species, they undergo very stereotypical behavioral and reproductive cycles, and that will become important later in this talk and also in the talk this afternoon. What they do is they go back and forth between what we call reproductive phases, in which the queen lays a lot of eggs, and then the colony doesn't migrate. That lasts for about 3 weeks. And then when the larvae hatch, the colony switches to what we call the brood care phase or the nomadic phase. And in that phase, which lasts for about 2 weeks, the colony has larvae and the ants migrate almost every night to a new nest site. So it's this very kind of like an idiosyncratic kind of lifestyle, right? It's like it's a little bit like the circadian rhythms maybe that Jeannie mentioned, but it's on a much larger scale. And I'll talk a little bit about how that works in the second talk this afternoon. So now if you sit there at this emigration column, you watch this like stream of ants walk past you. So these colonies can be massive. They can be many hundreds of thousands of ants. And these emigrations, they last for many hours. And of course, but what's nice about this is the whole colony passes past you. So you can see, by just watching this immigration, you can see anything that's in this colony. if you pay close attention. But if you do this, you get this kind of hallucinations at some point. You close your eyes and you see like ants just crossing your visual field. It's terrible. Sometimes these emigrations happen at night, and sometimes we go back to our research hut at like 3 in the morning or 4 in the morning, and I try to fall asleep and I just see ants moving in front of my eyes for like another hour or so. This is one of the special events during one of these emigrations. This is when the queen comes by. So there's one queen in each of these colonies. And you can see her walking here. In the actual emigration, it's really difficult to see her. That's why I've taken her out here and put her on a white background and photographed her. Because she's always covered by this retinue. So she has this like queen retinue of ants. And you can see that it's mostly these like large ants that have these big mandibles, the soldier ants. That I also showed you on this picture of the 6 different species of eceton at La Selva that are with the queen and are protecting her. And they're basically covering her more or less completely. She walks independently in the immigration, but they're mostly covering her. But so in the second project that I'm going to talk to you about today is we're sitting at these nightly emigrations. So every night, somewhere in the middle of nowhere in the rainforest in Costa Rica, me and Christoph mostly. And we were watching these emigrations. And so you see the stream of ants, thousands of ants. And every now and then, there's like some weird thing walking past, right? Looks completely different. This is a beetle larva. There's a beetle larva walking in this trail of ants. And the beetle larva is following this trail pheromone that the ants deposit, and the ants don't notice it. It's like really integrated into the ant colony. It's like a really strange thing. And this is one of these social parasites. So there are many species that live in ant colonies, especially ant colonies in species that have very large colonies. And the reason is that, of course, these ant colonies are very well defended, but once you somehow crack this defensive code of the ant colonies and you manage to get inside, they are very, very resourceful, right? There's always a lot of food inside ant colonies. They're ant larvae. It's very well protected against other predators. So it's really nice to live inside an ant colony once you manage to get in there and the ants kind of accept you as their own. And so especially in army ants, there's a lot of species that live inside these ant colonies. And so it's so diverse that it's really a little bit of a microcosm in itself. And so we wanted to study now not the prey of the ants, but the parasite of the ants in a very systematic way. So the second part of the talk is looking at army ants in this host-parasite interaction network. So we're first interested, of course, in a very basic question: how many species of social parasites are there to begin with? And this project, we really only focus on these 6 eceton species at La Selva Biological Space Station, because those have the emigrations above ground, so we can sample everything that's inside these colonies. All the other species, they usually emigrate below ground, so it's very difficult to collect their social parasites, even though they also have social parasites. And again, we were interested in how specialized are these social parasites. Do they parasitize one of these army ant species? Are they very specialized on this one, and do they have particular adaptations to this one species? Or are they broadly just army ant parasites that you can find in any army ant species? And then the question is, how have they co-evolved with the ants? What are their kind of specific adaptations to live inside ant colonies? How have they cracked this defense code of the ants? And are there general patterns how generalist social parasites differ from specialist social parasites? And again, we used a similar approach to what I showed you before, the systematic sampling followed by DNA barcoding. And so this is just a video of what this looks like. This is towards the end of a colony emigration, so the traffic is not that strong anymore, but you can still see the ants run past. And every now and then you see this green rubber tube that comes in at the top. This is actually Christoph with what we call an aspirator. So there's one tube that feeds into a little collection vial, and then there's another tube that Christoph has in his mouth, and he's going to aspirate. And then there's an airflow that sucks in little insects into this tube. It's really important. See, and now there's one of these guys coming. Whoops, and here goes. It's really important that you hold the aspirator the right way. If you sit there in the middle of the forest at night, and after 5 hours, the chances are at some point you'll flip the 2 tubes, and you get all the stuff that you aspirate right in your mouth. And it's happened to everybody more than once. It's particularly painful if you just collected this one thing that you've been waiting for like 5 minutes, and 5 hours, and suddenly it's in your mouth. So this is how we collect the army ants. And if you're very good at the army ant parasites, and if at some point you become very good at this, you see that Christoph really didn't disturb the ants much. The flow just continued. And so you can really sit there, and in some ways it becomes meditative. So as a first example, if you do this in a very systematic way, we discovered a few really extremely charismatic new species of social parasites that weren't known to science at all. So this is my favorite example. This is a parasitic beetle that attaches itself with its mandibles to this to the pediole, which is this intermediate part between the gaster of the ant and the thorax of the ant, and has these retractable legs, so the ants really there's no point of attack. The ants really can't grab the beetle with their mandibles or bite it. So here's the gaster of the ant, and this is the beetle. This beetle species is actually Christoph named after me, so I'm very glad for that. Now I have an ant butt beetle named after me. It's kind of a nice honor. And when we describe the species taxonomically, of course, you have to provide a very detailed description. So usually we team up with specialists. Neither Christoph or me are taxonomists. But so we team up with specialists here. Christoph teamed up with Alexei Tishechkin from the Smithsonian in Washington, who's a specialist for this particular group of beetles. And that beetle became quite famous. It was like featured on like TV shows and I don't know, the radio. And it was elected as one of the 10 new species, top 10 new species of 2018, I think, together with some like large extinct mammals and an orangutan species. So it was got a lot of press coverage. There's a children's book that a former Ph.D. student of mine just sent me a couple of weeks ago that came out last week where this beetle is featured. So this is just a very charismatic species, right? Like it rides on the ants and has this really cool biology, and it's completely new. Like there was nothing similar known before. But with this DNA barcode approach, actually most of the new species that we discovered are what is called cryptic species. So when you see them, they look like known species because their differences are so subtle that it's even for a taxonomist, usually they don't recognize them as separate species. But of course, if you're really interested in how specific interactions are between species in an ecological network, you have to have very fine resolution of where the species boundaries are, right? So what had happened in this case, this is a Vatesus beetle, and those beetles also walk in the emigrations of the ants. They look very different, right? They have these kind of drop-shaped body forms. They also have retractable legs. So again, they're very difficult for the ants to grab. But in this case, we started to sample these beetles, and initially we thought that there's only two species. One species of Vatesus that parasitizes these two aceton amiant species, and then we thought there's a second one that parasitizes these three species here. But then we started our DNA barcoding approach, and once you start to have all these different barcodes, you can cluster them in genetic clusters, in genetic groups by similarity. And clearly these two here, they form very distinct clusters, right? But then there were two additional clusters that became very obvious. These two here, we call them 1 and 2 for now. And what was striking is that this one cluster you found only in this one host species, and this other cluster you only found in these two host species. So that's a pretty strong sign that these are actually not the same species, right? Because why, if you had one species, why would one genotype always end up in one host species and one genotype always end up in the other species? But you can do another trick. You can look at a different part of the genome that is independent from this part of the genome. This is a mitochondrial gene. So we looked at a second gene and actually a few more genes that are nuclear genes. And those are, they get broken up during recombination, during reproduction. And so they should be inherited independently. And they should basically mix. But if you look at this nuclear gene, you see the divergence is much less. Nuclear genes evolve slower than mitochondrial genes. But you can still see that these clusters completely hold up. This cluster here of the mitochondrial genes only ever has nuclear genes from this cluster of the nuclear genes. And this cluster of mitochondrial genes always has nuclear genes from this cluster. So very clearly, there's no mixing and matching between these two clades, between the nuclear and the mitochondrial genes. And that's really important from a biological perspective, because the way we define species usually is by the fact that they're reproductively isolated, that they can't interbreed, they can't mate and produce fertile offspring. But that's often, in reality, very difficult to decide whether that's the case, but genetically you can do it. And we know that if these clusters, if beetles from these two clusters mated with each other, we would see reshuffling between these two clades, right? So we immediately know from this data that these two beetle species are actually reproductively isolated. So they're genetically distinct, and they're different species, and it means, of course, now that there's not one beetle species that can infect 3 different host species, but there are 2 beetle species that each infect only 1 or 2 species. So that makes a big difference when you start to think about specificity in species interactions within this ecological network. So in total, we looked at over 2,000 of these parasite species from these 6 amiants that I told you about. Most of the parasites in this particular network were beetles, and some were forward flies, these wingless forward flies that you can also then see walking in the immigration columns. There were also some other cool things. There's a silverfish, and there's even a millipede that walks in these immigration columns. In some army ant species, not in this community, but in others, there's even snakes that are myrmecophiles, that are social parasites. They feed on the ant brood, and you can see the snake walk or slither in the immigration column. And now, again, you can plot this in a network. Here, I've plotted this a little bit differently, but here are the 6 army ant species, the rows of this table. And then there are the 61 different social parasite species in the columns. And the shading of each of these squares indicates how frequent they are associated with any particular species, like in what proportions of colonies that we sampled you could find them. And you can see that there's some species that are very, very generalist and very frequent, like the silverfish, for example. You can find it in any army ant species you look at, and there's really only one species of silverfish. Then there are other species that are very, very prominent, so you can find them in any colony of that particular species you look at. They're always there, but they have a much more narrow host range, like this Vatesis beetle that I showed you here, only infects 2 species. And then there are others, like this army ant bud beetle that I showed you in the beginning, that's very, very specialist. You can only find it in one particular army ant species, and even in that species, you don't find it in every single colony. And that's probably part of why that species wasn't known yet, right? You really have to do a lot of effort and systematic sampling until you find something like this. So this specificity between the different types of parasites is highly variable. And now we can, this is, well, we'll see how it shows up. You can compare the specificity now of such an interaction network. You can calculate different types of specificity indices, and you can compare different types of networks. And here I've plotted 2 types of networks. This is a network between ants and plants. This example that I showed you in the very beginning of my talk. These are very specific networks, highly specific interaction networks. And you can see that the specificity index of these kind of networks is somewhere on the order of 0.8 on a scale from 0 to 1. So these are highly specific networks. There's always one species and one plant, and very little switching between the different mutualists. This is a much more loosely connected interaction network. These are ants that feed on what's called extrafloral nectaries. So a lot of trees, they just present nectar at these little openings extraflorally, so not in flowers. And then that attracts ants, and the ants kind of loosely defend the tree, but it attracts all kinds of different ants. So it's a very highly connected non-modular interaction network. And that's reflected in this overall metric of network specificity. Now, if we look at where the army ant parasite interaction network falls, it's somewhere in the middle. So it's kind of an intermediate level of specificity. But what's important, again, is that the specificity, once you start to look between different groups of parasite, it actually varies widely. So there's some groups of parasites, some types of parasites that are very specific, and others that are very non-specific. Just for comparison, this is what it looks like when I plot the army ant prey interaction network. That's much more specialized, or much more, the specificity is much higher than in this army ant parasite interaction network. But you can use this approach of DNR barcodes to get even more insights into the biology of the species, just a little bit like what we did with the interaction network between army ants and prey. So this is this weird-looking thing that I showed you in this video clip that walks in the army ant immigration columns. This is a beetle larva. So now with the DNA barcoding, just like with the prey items in the beginning, you can ask what kind of adult does this larva belong to, and it turns out that this is actually the larva from these Vatesis beetles that we looked at. And now we can ask questions as to when in the colony cycle of this host species do we see larvae, and at what developmental stage are the larvae? When do these beetles start to lay eggs, and how are they synchronized with the reproductive cycle of the ants? And it turns out that these beetles have actually evolved very, very sophisticated adaptations to this reproductive cycle of the ants that I described to you in the beginning. So I'll walk you through this a little bit heavy, dense slide in a little bit more detail. So in the middle, this is basically the reproductive cycle of the ants. And so they have this, what is called a stationary phase here, or the reproductive phase, where the queen starts to lay eggs. And then when the larvae hatch from the eggs, the whole colony shifts to this nomadic phase, or the brood care phase, when now they relocate the nest almost every night, or very regularly. And then when the larvae pupate, when they start metamorphosis, then the colony becomes stationary again, and the queen starts to activate her ovaries again and starts to lay eggs again. Now, it turns out that the beetles have a very similar reproductive cycle that's highly synchronized with what the ants are doing. So you can see that if you collect these beetles towards the very end of the stationary phase, you can see that now the females of these beetle species have developed eggs in the abdomen, and you already see a few of the very youngest larvae, the first larval insta, the first developmental stage of the larvae. And then as the colonies enter this nomadic phase, you see more and more like older and older larvae insta pop up. At some point, if you catch a colony that's somewhere in the middle of this nomadic phase, all of the beetle larvae that you'll sample are going to be in the last larval instar. And then at some point, they drop out. If you collect the colony or sample it from a colony like this in the late nomadic stage, there are no more beetle larvae in there. And from that, we conclude that at some point, the beetle larvae leave the colony, and they probably undergo pupation somewhere in the leaf litter. They disappear, and then the adults have to fly, and they have to find new colonies. But what it means is that these beetle species have really evolved very complex life history adaptations to live with these army ants. And I'll give you another example where we can kind of contrast two types of beetle. And these are both staphylinid beetles, rove beetles. There's one group in the genus Tetradonia, and these are usually generalists, and I'm talking now in terms of the host spectrum. So you can see that each species of these Tetradonia beetles, they are found with many different ant species, at least most of them. Some only with 2, some with 3, but some also with all 6 of them. But there's another type of beetle that is also a rove beetle, so not like fundamentally different. But these beetles, you always only find them associated with a single ant species. So one group that's pretty broad, and one group that's highly specialized in terms of their host spectra. And now if you look at the biology of this species, it turns out that the biology is also very generalized for these ecological generalists. So the body plan of these rove beetles are very typical rove beetle-like. They don't usually interact with the ants. You can do behavioral interaction assays in the lab. They always hide from the ants. They don't want to be in touch with the ants. They don't walk inside the main column of the immigration. They walk usually in the periphery, or sometimes they walk past in the immigration once all the ants have passed. And what these guys do is they prey on old and dying ants, and you see them at the periphery of the immigration column or the bivouac of the ants, and then they drag all the dying ants into the leaf litter where they feed on the ants. So very generalized predator. This other group of beetles, they're very, very specialized. So you constantly see them interact with the ants. They groom the ants. They have evolved these, what people call myrmecoid body form. So they kind of look like the ants. They mimic the color of the ants to some extent. You see this here. They have really like the head and the thorax is very similar in color to the ants. They walk in the immigration columns. This is actually a raiding column. You can even see them here crossing one of these bridges. So they're constantly in contact with the ant. They live inside the ant colony. They're highly integrated. And if you do chemical analysis, and that will become more important in my afternoon lecture, where I'll talk about how ants actually communicate, you will find that these specialized parasites, they have kind of cracked this chemical code that the ants use to tell self from non-self, or foreigners from colony members. These generalist beetles, they smell very different, whereas these specialist beetles, they really smell like the ants. And this is an analysis where we basically we just extract surface chemicals from the cuticle of the ants and the beetles, and then you can analyze them with gas chromatography. And you can plot them in this kind of principal component analysis, so it gives you kind of a multidimensional chemical space, but you can statistically analyze it in a way that you can project it to only 2 dimensions, so you can visualize it nicely. And you can see that essentially just from this chemical profile, it wouldn't be possible to say whether a chemical profile comes from a specialist beetle or from one of the host ants. But it's almost always possible for these generalist beetles. They smell very different from the host ants. Okay, so what I've shown you here in the second part of the talk is that social parasites are very diverse, and they target all army ant species. Every army ant species that you look at will have some of these social parasites. But there's a wide range in how specialized they are. Some of these social parasites are very generalist. Some of them are very specialist. And many of these specialists have evolved very sophisticated adaptations to life with the ants, whereas the generalists usually don't. And again, you could ask similar questions as to what I asked for the prey for the predator-prey interaction networks. What happens if you move now to a different site where you have a different composition of army ants, for example? Do you now see that certain social parasites have a different host spectrum? Right? Like, are some parasites excluded from certain ant species at La Selva due to competition with other social parasites, for example? These kind of questions that then allow you to get more general information about what structures these interaction networks between the host and the parasites. Okay, so now to get back to this initial question as to what are some of these principles that govern life. And of course, as I said, there's a lot of these principles. The more you learn about biology, the more you start to see principles everywhere you look, basically. But I'll just point out a few things that are worth thinking about, I think. So I've shown you that some species and ecosystems are what we call keystone species. And they're called keystone species because they really tie things together and they have a completely disproportionate impact on the ecosystem as a whole. And I've shown you this one army ant, S. tombuchellii, as an example. You can easily see if you would remove that species from an ecosystem, the whole structure of the ecosystem would change dramatically. And that's interesting from a network perspective because these keystone species, they really function like hubs in an ecological interaction network. And again, these kind of hubs you have in things like neural networks, for example. You have hub neurons, or you have them in metabolic networks, and so on. So these are very generalized features of complex interaction networks between biological units. What I've also shown you is that there's often really strong indirect effects in ecological networks. And so, of course, I've mostly talked about these direct networks, these direct effects, because they're easier to measure. But you can easily imagine how the strong effects that something like an army ant has on the ant-prey community, or even on the social parasite community, has then cascading downstream effects on other organisms as well. And so you can get these interaction effects that provide negative feedback or positive feedback between certain types of organisms in one of these ecological interaction networks. And of course, you see similar feedback loops in things like neural networks or metabolic networks in a cell. I've also shown you that competition is often decreased by specialization, both in the parasite example and also in the predator-prey interaction network example. So one of the reasons that you can pack so many species in an ecological network is that these species are often very specialized on certain ecological niches. And that seems to be like an overall evolutionary trend. And in the last part, when I talked about these generalist and specialist social parasites, I've shown you that often this ecological specialization and this evolutionary specialization, where a species evolves very specific adaptations to live inside, in this case, a particular host ant species, they often go hand in hand. And one idea behind this is that there's a trade-off, right? If you want to parasitize many species, it buffers you against, say, the risk of one of these species going extinct. You still do fine because you can live with other species. But it doesn't really allow you to specialize on one species because you have to remain flexible, right? You have to be able to also colonize other species and interact with other species. And that kind of limits how specialized you can become on any particular species. All right. So that was the broader messages from this morning talk. And again, I'm happy you're all here, and I hope you have fun over lunch. And we have an ant booth and a little ant exhibit somewhere that I'm sure you'll be guided to. And then I'll see you again this afternoon. And if you have any questions, you can ask me questions.
Hi. So they're bringing back a lot of insect material back to the bivouac, right? And so that'd be a lot of chitin. Where does all that go? Because when they pick up and move, do they leave it behind? What are they doing with it?
So these army ant bivouacs have kind of a garbage heap that's like below the bivouac. And so they drop all the food remains on that garbage heap, and dead ants as well. And that garbage heap of army ants is actually a whole ecosystem in and by itself. So that could be a whole another story, because there's actually a lot of scavengers that come and live inside this garbage heap. So it's another very biodiverse ecosystem once you start to look at it. But that's how they do it. And then of course at least during the nomadic phase, they move during the day, so there's only a small garbage heap. But if you look at the bivouac during this phase where they don't move the colony every night, these garbage heaps can actually become quite large and kind of impressive. They're very good at keeping the nest itself clean, though. Okay.
Hi, first off, thank you. It was really, really cool.
Thanks.
But since the army ants, go and take out everything in their path, I was wondering if you had any examples of army ants fighting other army ants. Because I figured, that might be a thing. So have you seen any of that? And if you haven't, do you have any reasons why?
So interestingly, there's no examples of army ants regularly preying on other army ants. There's very, very few examples, I think, and it's all very anecdotal, where actually conflict escalates between different army ant species. So usually what you see is that, for example, at night, when they emigrate, when they move the nest, sometimes you can have paths of two different army ant species cross. And so there's these little, there's a little bit of fighting in the periphery, but usually there's just like a standoff. They like recruit soldiers to the area where they cross, but then they usually manage to keep it kind of peaceful, and it never really escalates. And one idea is that these army ant colonies, they're so huge and kind of specialized on fighting that if you, as an army ant colony, you attack another army ant colony, it's just it's going to be an extremely costly interaction, right? Like there's going to be massive mortality on both sides, basically. And that's probably one of the reasons why it's not a good strategy as an army ant to attack other army ants. They're just too big.
So that, I'm sure it's several species, but the ones that build a nest, but it's just entirely made of army ants, and then they relocate every night. What kind of range are they covering, like, let's say, within a week, if they're doing that every single night?
So the emigrations are usually somewhere around 100 meters, sometimes even a little bit more. And that's actually a lot, like if you think about the fact that they are ants, right? And even from a human perspective, if you, in the middle of the night, you don't see anything, you have to find your path through the rainforest over 100 meters, is actually quite long. Sometimes they emigrate like 150 meters or something, and there's no path, so you have to like walk through, and they go down cliffs, and they don't care, right? And they do that, there's a little bit of a controversy about that. Some people say that they emigrate, the subsequent emigrations kind of extend in the same directions. What they do is they emigrate in the direction of the swarm rate during the day. It's a little bit unclear whether they go in the same direction, whether they kind of keep a compass bearing throughout this nomadic phase. But either way, so there's the nomadic phase, maybe they do like 10 emigrations, so they can easily emigrate over a kilometer each month, right? And the emigrations here are different from like migrating birds or something, right? Like migrations, animals go somewhere and then they come back in a very predictable way. And army ants, they don't migrate, they emigrate, right? So they just leave, and you'll probably never see them again. So they can, over the lifetime of a colony, they can probably cover very large distances.
I noticed in the satellite image of the rainforest, and there were leafcutter ants, and then there were like white patches. I was wondering what exactly we were looking at. Are they like entire trees covered with fungus, or is it like 5 trees in a row, or like how many trees are they covering, I guess, my question.
So this is mostly, these white patches are barren soil, which is where the leafcutter ants just cut everything down.
Okay.
So they just remove the vegetation, basically, right? And yeah.
Okay, thank you.
When you said the social parasites, the beetles, had a way of tucking in their legs to avoid the mandibles, do the larvae have a similar mechanism to avoid being eaten by the army ants?
Where are you? I can't.
Right here, right here.
Okay.
Sorry.
I lost track of the microphones. So for this particular beetle species, we actually don't know the larvae yet. We have never found the larvae. And that's actually true for a lot of these social parasites, where for some of them, like these Vatesis beetles, we know, because the larvae are kind of obvious and they walk in the emigrations. But what we think is going on for a lot of these beetle species, the larvae, maybe they develop in these garbage heaps that I described before, or they just lay their eggs somewhere in the leaf litter, and then the larvae actually develop outside of the ant colonies, and then the adults recolonize the ant colonies. So for that particular species, we don't know. So, and of course, if the life cycle is not synced with the ants throughout, if only the adult stage, say, lives inside the ant colonies, then the immatures don't necessarily have to be defended against the ants, right? So that's probably what's happening with this particular beetle. Because otherwise, we've tried to survey all kinds of immature stages we found, both in the garbage heap, at least a little bit, and inside the bivouac, but we've never seen, through our barcoding efforts, we've never seen an immature stage of that particular beetle. So I guess they're not there. That's my best guess.
Is there something intrinsically different about the beetles in the colony, about the ants in the colony that form the scaffold or the bridge versus the ones that climb over them? And do they switch places?
So there can be like division of labor between tasks, like the different ants specialize on different things, and that's something I'll talk about in the afternoon more. In that particular case, with the bridge forming, I don't think they're specialized bridge formers, but what happens is that, like say the raiding trail comes to a crevice, and then usually what happens is that some ant, like the first ant that comes, just walks so fast that it falls into that crevice, but holds onto the rim. And then the second ants will step on that ant. And one of the rules, so these kind of emergent properties in architecture, like of the ants, they kind of arise from very localized interactions. So as long as ants keep stepping on your back, you stay in place. That's one of the rules. And so these bridges basically maintain, and these chains maintain, as long as there's tension on the chain, the ants stay in place. And that can be for hours, right? So once this bridge forms, the ants that form the bridge, they will stay there until the traffic ceases, and then they'll pop out and then they walk home. But they can be in this bridge for several hours. As long as the traffic flow is high enough, they'll just remain in place. But which ants exactly form the bridges, I think is more a function of who falls into the hole first.
When the ants are traveling across or immigrating, when they reach a deep river, what would the army ants do?
When they reach a river?
Yeah, and they want to cross it.
So there's actually, when they reach like very small rivers or like small streams, in some cases they actually can't cross it via like these little bridges. And often there's like branches that hang across the stream. So on several occasions, when I was following emigrations at night, I actually had to walk through small streams. And then usually the ants, of course, they kind of can bridge small gaps with these bridges that I showed you, but then they usually find ways to go via stones and branches, or like a fallen tree trunk that crosses the river. Like in the rainforest, that stuff is everywhere, right? Large rivers, they can't cross. So actually, we did some population genetic studies where you look at the structure of like genetic diversity within army ant populations. And you can see that rivers are very strong barriers to dispersal for army ants. And the reason is that these queens that the army ants have, they're never winged. So they're not like many other ant queens that can fly. They are born without wings. They never leave the colony, so they always have to walk. So army ants only disperse by walking. And so even if there's a river that's wide enough so that a single fallen tree is not going to be able to bridge it, then the army ants are not going to cross it. And that's actually reflected in the genetic structure of the populations.
So what is the most aggressive species of army ant? And also, are male or female ones more aggressive?
Okay, second question first. So all of the ants I've shown you today, they're all female. Like if you look at an ant colony, usually all the ants in there that you'll see are female. All the ants that raid your sugar bowl, they're female. The ants that you see on the sidewalks of Manhattan doing these little tournaments in the spring, they're all female. All the ants that fight are always female. Like the males are only produced kind of sporadically, and they basically only serve the function of reproduction. So they mate with the queen, and then they usually die. So they're very transient. It's kind of the life as a male ant is kind of sad. So these are essentially all female societies. All the social behavior is performed by females. All the soldier ants are female. All of these guys are female. The most aggressive army ant. That depends, I would say, on who you are, right? Like if you are a specific ant species in the rainforest, you'll say that this one army ant that comes by every few months and kills out my colony, that's the most aggressive army ant, right? As a human, the most, there's very kind of exaggerated like accounts of army ant attacks in the literature. But I would say the kind of scientifically credible accounts, the most impressive ones actually come from African army ants that I haven't shown, like these African driver ants. In some ways, they're similar, they're related, but the colonies are even larger. So these colonies can be 10 million ants strong or something. And they're actually accounts where like an army ant colony overnight stripped the chained crocodile to the bones. So these can be very aggressive. And there's also accounts of again, these are anecdotal, and it's always a little bit, you have to be careful, but there's quite credible accounts where people left, say, like a baby in the yard and it was killed by army ants. So these things can happen. So I would say if you want to go for most aggressive, I would probably go with these African driver ants.
Hi. Oh wait, is it on? Okay, it is. Hi. So in terms of the social parasites, I was wondering if they're ever rejected from the ant colonies, and if they are, what that rejection process is like, and if them being accepted or rejected is more common.
So I think it depends on the parasite species. As I showed you in this last example, right, where I contrasted the specialist beetles to the generalist beetles. So the specialist beetles, you never really see them being rejected. They're so well integrated, the ants, they kind of treat them as one of their own. For the generalist beetles, they are aggressed. Like if you take a generalist beetle and you put it in the middle of a clump of ants, they'll actually aggress it. So the beetle is very careful not to get too close to the ants. So they kind of always stay in the periphery of the colony, and they only dart in briefly, grab, maybe try to snatch an ant to eat, and they're out again. So it really depends on the species. So these generalists, they are often aggressed, but the specialists hardly ever are.
I have a question about a parasite that wasn't mentioned, and it's actually a fungus, cordyceps, and I'm curious to how cordyceps affects the colony. Because
so cordyceps, it's one of these, maybe some of you have seen pictures of it. It's this fungus that induces the famous zombie ant behavior, where ants wander off from the colony and then they kind of bite on a plant, and then the fungus grows out from the back of the ants. It looks kind of spectacular, and it's one of these like mind-altering behavior hijacking fungi that affects ants. So that's how it affects the ants. So cordyceps is a little bit different in the sense that I don't think about it as a social parasite. Because what cordyceps does, it doesn't infect the colony, it affects individual ants. So cordyceps, it's very common, and it's a common insect parasite, right? You can see, like cicadas are often infected by cordyceps. All kinds of insects are infected by cordyceps, not only ants. So they don't really take advantage of the colony resources per se. They don't really infiltrate the colony in the same sense as these social parasites do that I showed you here. What they do is they infect individual ants, and then it changes the behavior of the individual ants. They just leave the colony, and then they die outside of the colony. So it's a slightly different type of parasite. You can ask me more questions if you want. All right.